This is episode 115 of the Immunology Podcast, Intercellular Communication with Dr. Harikesh Wong. Hey everyone, this is Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, I have Dr. Harikesh Wong from the Ragon Institute and MIT on the podcast to talk about his research on how cells assemble and communicate to control immune responses in tissues. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
STEMCELL Technologies would like to introduce you to Immune Regulations News, a free biweekly newsletter brought to you by the STEMCELL Science News Program, covering research on the regulation, suppression, and modulation of the immune system. Immune Regulation News keeps readers current with the latest news, research, policy, events, and jobs relevant to the immunology community. Subscribe for free at www.immuneregulationnews.com.
all right.
What up, Jason? Ready for another another academic year?
Yep, the kids are back in school.
Nice. Some space at home now.
Yes, during the day.
So, Jason, if you were to go back to school, would you do something? Would you study something else?
I can't go back to school. I've done too much.
You did too much.
What would you do?
That's fine.
You've had
what would I do?
So if you had to go back for another couple.
Oof. Well, it depends. If it were starting over and doing school again, maybe, I don't know, I like it. Maybe engineering. Sometimes I wish I could do more relevant things, like, I don't know, how the world works, not just the little tiny cells in my body. And if I had to go back now, I don't know, something completely different. Become a diplomat. I don't know, something completely different.
You could be a diplomat. You could speak all the languages at the UN.
Yeah, at least somebody would appreciate that, right? That would be useful. Yeah, no, I think I'm good, but that would be fun.
You don't want to translate things that then just got shot down every time on final vote by everybody, by a different group every time.
Yeah, that sounds that sounds very frustrating. No, I'm okay. I'm good. All right. So, I mean, we keep learning, right? Every week we're learning a new paper.
We are. I like that segue. We're all students of science. All right.
We're all students of science.
I have one from the Ragon Institute and MIT, which is the same place as where we are having our guest from.
What a coincidence. What a productive institute. Okay, shoot.
All right. It is in Cell. Came out on September 11th. Immunometabolic deficits of CD8 positive T cells disrupt gut barrier integrity in people with HIV. First author is Upasana Das Adhikari, and last author is Douglas S. Kwan. This actually is a really quick paper to describe, even though they use pretty sophisticated methods. So, there is a known problem where people with HIV have leaky guts and gut inflammation, and like there's some gut dysfunction even when they're on highly active retroviral therapy and basically doing well, right? It's like a chronic problem from the infection, not in your CD4 cells, right? Like it's like, what is this? It's weird. But if you remember, we've seen HIV infects not just those cells, affects all T cells, affects macrophages, does a lot of things, right? Even if you're keeping your cell level count high, it's still a retrovirus. It's still there. Dun, dun, dun. So they're like, this is weird. So they want to understand what was happening in this case. And so they used intestinal fatty acid binding protein and found that there was indeed higher levels of this in people with HIV who were either antiretroviral naive or treated compared to uninfected individuals. And this was kind of where they started this. They took colon organoids and studied them in patients from them and found that the primary organoids had repair deficits, but once you passaged them, the effect went away. So it was something that went away on secondary passage. And then they found in the passage, there were T cells there initially. So they did flow and looked and like, there's CD45 cells here on the first set. But if you got rid of those, then the weird effects stop happening. So they cultured secondary organoids with people with immune populations from people with HIV to figure out what was doing it, and CD8 cells, positive T cells, were the culprit. Dun, dun, dun. And then they looked at like, well, this is weird. Why are CD8 memory? And it was CD8, mostly the memory effect cells that were doing this, were being naughty. And they're like, well, what's going on here? And they looked at it and they had altered lipid metabolism and had trouble doing lipid metabolism, getting energy from it. But they need it. And there was decrease in PPAR gamma. And so there's all this dysregulation of the lipid metabolism. I'm like, well, okay, well, how the hell does lipid metabolism affect the epithelial cells? This is lipid metabolism in the immune cells, right? Like, but the epithelial cells are getting the damage. So they do a bunch of work and try to really understand what's happening with fatty acid oxidation. If you do a PPAR agonist, it fixes the problem. And it's like, oh, there's PPAR dysregulation. And they then look at like they look at like, so they integrate lipid into the lipid bilayer of epithelial organoids, a dye. And then the dye shows up in the CD8 cells that are co-cultured with it. And what they eventually establish is these dysregulated CD8 cells are so hungry for lipids and so broken inside, they're eating the epithelial cells and pulling the lipids from them, causing the damage which causes disrupted barrier. And now I'm done. And it's PPAR gamma signaling is required to maintain the barriers. When you break PPAR gamma down, you have more lipid scavenging versus standard pathways. They have to scavenge more. And so this extra scavenging, they start scavenging from their buddies, the enterocytes, these greedy ass cells, and damage them. And that's why they have a broken barrier. And that's why the primary organoids, which have some residual CD8 cells, don't grow well. But when you culture them and just get the second passage where everything's mono-associated and you get single cells, those organoids are just fine because they got rid of the asshole CD8 cells. It's so cool. It's so wacky.
Man, that's like the call is coming from within. It's your own cells that are, eating up the cells they shouldn't be messing with.
Yep, exactly.
So these are people that are controlling their viral load with the antiretroviral, but still this.
It doesn't matter if it's controlled or not. Either way, the problem's there.
Yeah, and this is because the HIV has integrated in the cells.
Correct.
And that's what's kind of giving these issues.
Yeah, it's the cells that are the problem.
Yeah, yeah. Cool.
Isn't that wild?
Yeah, it is.
I really like this paper. Like, I was like reading it. I'm like, huh, okay, why do I care? Oh my God, they're eating the cell. It's like the greatest turn at the end. I loved it. I loved it so much.
They're like, oh, what, figure 7. All right. Like, what?
Cells.
Wait a minute. Well, is the invasion of the zombie T cells.
They're literally eating. They put the dye in. It was such a sophisticated thing. They put the lipid dye into the intercalated into the epithelial cells and watched it get nibbled on and eaten into the lipid.
But how is that
causes it to undergo apoptosis? It stresses the cells out when they get chewed and they start dying.
Yeah. But what are they like? What are the neutrophils? What is wrong with these T cells?
The T cells can't manage their lipid metabolism normally, so they have to scavenge more to get all to push the pathway because the pathway is broken.
But still, are there any kind of mechanistic things that need to change in order for T cells to be able to do this? Because it doesn't feel like something that T cells.
They normally scavenge lipids. The problem is that their lipid metabolism is broken, so the scavenging is turned on because it can't do it normally. So if you agonize, if you turn PPAR gamma on with drugs more, it works just fine. But the PPAR gamma driven lipid metabolism is all screwed up.
All right. Nice turn of events at the end. Very interesting. All right. So for my first paper of the day, I have also something that happens when T cells are being, stressed out and they're doing things to them are happening that they shouldn't be happening. So the paper is called Oxidative Stress Induced Telomere Instability Drives T Cell Dysfunction in Cancer, published in Immunity in the future. So they have this thing which I guess is published ahead of press. So it's on the October 14th issue. Obviously not that hasn't happened yet. Well, that's how it works for this journal. And first author is Diana Rivadeneira. And this is from corresponding authors Patricia Opresco and Greg Delgoff at the University of Pittsburgh. And basically in a study that they're looking at, reactive reactive oxygen species, which are a big issue. That we're studying it all over the place. Reactive oxygen species are not a good thing. They will damage things in cells. Uh, and in particular, they have many ways by this oxidative stress can affect DNA. You have these things, oxidized DNA bases, in which you have a conversion, the bases get oxidized. And then there's specific bases that are more likely or more susceptible. And in particular, guanine is very susceptible to this. You can have a basic sites in which basically the base, the amine base is removed from the is separated from the phospho backbone, or you have deamination, in which, for example, a cytosine can be changed into a uracil. So all of these things are bad. They will mess up with your DNA, and these are things that the cells have evolved many mechanisms to keep at bay. And here comes T cells. And T cells in uh, tumor microenvironments, we know that they are subjected to very unfavorable conditions. And I think it's kind of non um, well known that one of the things that happens is, for example, increase in ROS production in these metabolic stressed cells. And so in this paper, what they do is a bit the connection between this increased ROS production and the effects on the telomeres of the T cells, and how that has a direct effect on the survival and the kind of the uh, fitness of the cells in the tumor microenvironment. So, what we need to understand is that, for example, telomeres have these TTA-GGG repeats that are kind of part of their structure. And because there are so many G's in these repeats, they are particularly susceptible to what is known as this 8-oxo-G oxidation, one of the examples of oxidative stress on DNA. And so they make them particularly susceptible. So what they do in the study, which I think is pretty smart, they use this model that I was not aware of, which is a chemooptogenetic tool to generate singlet oxygen species, reactive oxygen species in kind of specific cellular compartments. So they have a situation in which they have a fluorogen activating peptide that you can transduce. You can this which sequence you can transduce, or you can generate mouse strains producing this that fuse to some other protein that will take this to a specific cell compartment. Um, and for example, if you want to direct it to the mitochondria, you can just uh, put a sorry, not a fusion, necessarily a fusion, but or a leading peptide. So they use the leading sequence of mitochondrial proteins, and that why you get this Fab peptide to the mitochondria. And once you have it there, if you add this dye called malachite green and a specific iodinated version of this dye, then it will provide an oxygen when the Fab is excited by a specific far red light, so 660 nanometers. So basically, what you can do is by having this construct in the right place and adding this dye, this modified dye, and shining a 660 nanometer light, you can generate ROS in a specific place, a very kind of specific location. There you can study the effect of ROS in specific compartments, which to is before without this tool it would be pretty hard because you can make ROS and then it goes everywhere. So you want to make it very specific. So they see that the first thing they try is they direct this to the mitochondria, and they show that if they make ROS in the mitochondria specifically, you have cells that have reduced proliferation. It shows signs of exhaustion, reducing production of inflammatory cytokines, interferon gamma, TNF, typical markers of exhaustion and or senescence, if you say loss of CD27. So this kind of overall unfavorable phenotype. And they show that when they look in, they induce this, and then they look at the telomeres in these T cells, and these are our mouse models. They show that there's accumulation of proteins that associate with kind of the DNA damage response, particularly these proteins called this phosphorylated histone 2 H2AX and this P53 binding protein, which P53 is a well-known downstream effector of damage response, the damage response. And they see this accumulation of these proteins at the telomeres, and they do all this fancy microscopy. But what they don't see is shortening of the telomeres. They don't see a reduction. They do fish to kind of measure the length of the telomeres, which will associate would be proportional to the strength of the signal from the fish. And basically, they see that there's no difference in the size, but there's increased damage that is observed through the recruitment of this damage response proteins. So far, that's what they see. And what's cool is that they can, they also look at this in kind of a tumor infiltrating mouse, so in mouse black 60 B16 ova models. And they check some human melanoma samples, and they say higher expression of these P53 binding proteins and the telomeres. And what's interesting is that although they cannot see reduction in the size of the telomeres in mice, they do see a trend to a reduced size in humans. And they're like, well, we're not sure why that is. Is that because human telomeres are already shorter than mouse ones? Or maybe because they cannot really have completely matched cells from, they have cells from melanoma and then from healthy donors. So maybe, they're not perfect control. So they cannot rule out that there's differences in the size of the telomeres in human samples, but definitely there's this increased recruitment of DNA damage response proteins. And what's cool is then they do well. So they say, well, probably the ROS is coming from the mitochondria affecting the telomeres. And they say, what if we actually make ROS at the telomeres? So they fuse this Fab protein to TRF1, which is part of the sheltering protein complex that is it protects the telomeres. So they really make ROS at the telomere site directly there. And this pretty much mimic much of the effect that they see from the mitochondrial ROS. And these exhaustion markers, senescence markers, reduce production of cytokines. So they think that they can show by this that indeed this telomere damage is kind of important for the overall loss of fitness of the cells. And they show that, so the mechanism of this is because of this very high susceptibility of telomeres to oxidative stress, because of the guanines that there are there, this is picked up by, the DNA response mechanisms of the cell, particularly activation of P53. And this is what drives the cells towards cell cycle arrest, and also seems to be part of the reasons behind the alteration of the metabolism and the function of these T cells, with eventually results in exhaustion, all these markers of loss of fitness of the cells. So what they show is that if ROS is the problem, can you get rid of ROS next to the telomeres and protect the telomeres from this ROS? So what they show is that they use a different protein, a ROS scavenger protein, it's glutathione peroxidase 1, and they fuse that to this TRF1 telomere, guiding protein. And they show that by doing this, they can protect the telomeres from this damage response that they see in TIL. So the idea is that TILs have damage in the telomeres because of their mitochondrial dysfunction, generates ROS that then goes and affects the telomeres. And they show that by protecting specifically the telomeres with this anti-ROS, enzyme, they can improve a lot of the exhaustion markers that they see. And in a way, this kind of shows that telomere damage is an important reason of why mitochondrial ROS affects the fitness of T cells. And when you look in vivo, they do some models with B16 tumors, and this improves the functionality in vivo of the CD8s and their presence in the tumor microenvironment, suggesting that this is one of the things that you can do to help kind of keep these T cells fit in their job against the tumor. So I really like these models. I thought it was pretty smart, pretty nice a way of looking at ROS very specifically. And I'm not surprised with the results, but it's nice to see this models to show it very carefully.
It's interesting. But so you're talking about ROS, but isn't cancer all about more glycolysis and less oxygen?
Yeah, but I think what happens is that you start having dysfunction of the mitochondria and uncoupling of and so that's where the ROS comes from. It's from sub from kind of mismatched metabolic programs that then you um end up having this ROS. Because, for example, you need to, even for glycolysis, you need to regenerate NAD somehow, and you might not be able to do it completely through a Warburg effect. So sometimes you might have this accumulation that might still drive mitochondrial function without actually making energy, and that might dissipate or might end up generating ROS. There's a lot of ways by which ROS can be generated. Not a lot, but it's well known that you have an increase of reactive oxygen species in in metabolic stress T cells in tumor microenvironment.
Well, I'm going to be talking about a completely different microenvironment. I have zero segue. It's also in cell though, so there's my linkage to the past, which was a Zelda pun. All right, this says microglia astrocyte crosstalk regulates synapse remodeling via Wnt signaling. First author is Travis E. Faust, and last author is Dory P. Schaefer. Now, why are we talking about neural signaling in an immunology podcast? Because microglia are fancy macrophages, right? So it's known that microglia can help remove synapses in some structures. During why do you want to remove a synapse? Neural pruning, lots of reasons. So apparently they have a whisker system where you pull whisker in a postnatal mice, and that in a specific part of the neuron in the brain causes there's no whisker anymore, so it remodels to get rid of those connections. Which I didn't know, but apparently you can just torture mice this way and do that and make them all go, whiskerless because it's a sensory organ. So that's the system they use. But they find in this case, it's not just the microglia, and they get something really interesting. And they first realize it's astrocytes are doing it. And so they do some really fancy microscopy, which is part of what they talk about their technology, is that they're looking at what the astrocytes are doing. And see, it's not just microglia, but astrocytes are getting close. And then the microglia remove the synapse. So the astrocytes reduce their association with the synapses first. And so that was the first finding. That's astrocyte first. It's not just microglia come in and then pop it. Then they do signaling analysis, see what's driving this. They do some single cell RNA-seq and all the other, and some co-immunoprecipitations for signaling, and realize it's Wnt signaling driving this. And then they look at which Wnt is doing, and they use this system where they blow out the they can expand the slide, make the tissue spread out more so you can see the space in between because the synapses are dense. But if you open it up, then the distance becomes bigger, and so you can actually measure things. And so they determined by ribosome affinity purification followed by RNA sequencing that with labeled ribosomes that it's Wnt receptors signaling causing the astrocytes to remove this. And so they map out that and figure out the TCF left pathway, and all of this is so it's okay, it's Wnt. They do a bunch of controls to show it's really the right cells they're seeing. With all their knockouts, they have to control some of it. And then they say, okay, if you activate Wnt signaling, does that work on its own? So exogenous Wnt works, hyperactivity with APC knockouts, which is a suppressor of Wnt beta-catenin signaling. It does it. Okay. Where's the Wnt coming from? So look at microglia cells with the same ribosome thing and see it's producing a ton of Wnt. And then they block secretion of the Wnt with a drug. And that stops the effect. And they then see, through RNA-scope, all this Wnt being produced, and then really go down and then knock out some Wnt and show that goes away. And then they're like, okay, so now the story is microglia produce Wnt that tell astrocytes to move the hell out of the way by reducing their synapses. The astrocytes do that, and then the macrophage goes and eats the synapse. Or not the macrophage, the microglia cell. That is a macrophage. Okay. And then to finish it off, they determine it's the CX3 CR1 system that's doing this, right? So that is the cytokine that's signaling, and they don't figure out how, but that's signaling the CXCL1, CXCR3 signaling is what causes the microglia upon damage. That is what's activated in the microglia that cause the Wnt, that causes the astrocyte, that causes it to all move. So that's the proximal driver of the whole thing. And so they map this all out with a bunch of tissue-specific knockouts and controls, because the knockouts aren't all perfect and have other cells involved, and then show that this is a CX3CL1 driven pathway that is activated, turns on Wnt in microglia. Wnt turns on signaling in astrocytes, causes them to move out of the way. So you see, cell cytoskeleton changes in gene expression. The astrocytes move out of the way, and the microglia gets rid of the synapse. So it's a kind of tour de force of signaling, mapping out that there's a lot of crosstalk back and forth. It's just not one thing, which is different than other cells.
Pretty complex, all this crosstalk between all the cells. So can you explain the idea of the whisker removal? It was just then they lose the connections that come from the whiskers, and that I don't understand how the whisker removal thing works.
So if you remove a whisker, right, that's like you be stabbing your eyeball out, and so it no longer works and can't come back. And
Right. Okay.
And that then causes the nerves to remodel, to remove the synapse in your brain for the connections of that whisker to the rest of your brain as a sensory organ.
All right. Okay. Yeah. 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 That sounds not very nice for the poor mice.
No.
But I guess it makes sense that it's the microglia that is actually doing the job, because that's what kind of
Right, but
macrophages do.
but in other cells they don't have the astrocyte component, which is weird. Like, whoa, the astrocytes are doing something here.
Right.
Oh, the microglia is still in charge because
Yeah.
macrophages are the boss.
Yeah, they are, aren't they?
Num 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 num.
Very nice. Very nice. All right. Do I have Well, I'm gonna be talking about myeloid cells, I guess. So that's the
That's a good
segue,
segue.
right? But then these are myeloid cells in the tumor. Bad myeloid cells that are, few at protecting tumors from the immune response. So the paper I'm going to talk about was published in Immunity, uh, oxidative. No, sorry, my bad. It was published in Nature, so we're changing providers. Myeloid progenitor dysregulation fuels immunosuppressive macrophages in tumors. And first author, Samarth Hedge, from the lab of Miriam Murad. So, from a friend of the podcast, Miriam Murad. And of course, makes sense that we're going to be talking about myeloid cells, specialty of the Murad lab. And basically, one of the big questions is, myeloid compartment is, it's well known that it's important for generating this tumor suppressive microenvironment. And the question is, what are the mechanisms that are, feeding this type of microenvironment? And so so basically in this paper, what they do is they do, a very detailed analysis of progenitor cells in the bone marrow of mice that have uh, this this is a tumor model that it's for small non-small cell lung cancer. And this is yeah, crass and P53 mice, knockout mice. And then they look very close into the hematopoietic compartment, stem cells, myeloid progenitors, monocyte progenitors, committed monocyte, all these different type of myeloid cells that I am still a bit confused by. But what they see is, they see an increase in certain in certain myeloid populations, and they focus on the ones that are kind of the main source of what are the suppressive monocyte-derived macrophages in tumors. These are um, this this GMP and CMOP. So those are the granulocytic monocytic progenitors and the committed monocyte progenitors, and they really focus on these ones. And one of the things that they see when they do all this analysis is they see an increased response to, well, I guess talking about Ross, the increased response to oxidative stress is one of the things that they see in this populations, and they really kind of focus on that. And they see transcription factors, and they focus on one signature, which is from the transcription factor NRF2, which is known for mediating the response to oxidative stress, and makes a lot of helps the expression of certain enzymes that will protect or will counteract ROS. And so what they show is that um, they see increased signature for this NRF2 uh, function. Also, they had some human samples, some human tumor tumor samples, and also look at the the myeloid cells in there, and they kind of really look into that, and they show that there is this increase in the ROS response, particularly in a NRF2 response in this my immunosuppressive myeloid cells, and also kind of low earlier in the development. So this um, progressive increase in this immunosuppressive cells that correlates with this protective phenotype. They see promotional correlations with these immunoregulatory modules, so that these suppressive phenotypes, and also dampened type 1 or type 3 interferon response, which I think these things were already known about these cells. And basically, what they suggest is that this activation of NRF2 is a consequence of these tumor initiated oxidative stress in the progenitors, and it's necessary for the progenitors to continue expanding. So in a way, it's kind of this myeloid cells, they have to respond to ROS, to this dysfunction that is generated because of the tumor, to continue generating cells. But in by doing this, they are upregulating this suppressive mechanisms, and this is what it's fueling their protection of the tumor. So it's kind of counterproductive response. And they do some tests, and they show that if you by knocking out certain genes in this pathway, they can actually rescue um, some of these inhibition of the anti-tumor response. And the other way around, if they eliminate KAP1, which is a protein that keeps NRF2 targeted to degradation. So by removing this enzyme, you have more NRF2. This actually uh, increase the size of the tumor, so works in the advantage of the tumor. And on the other hand, if you can inhibit NRF2, and they do this in some mouse models, you can improve infiltration of T cells, also natural killer cells mostly. So by doing this, I think they find that you have less cells, but also you have less immunosuppressive myeloid cells, which in the end it has a beneficial result on the anti-tumor response. So I guess this could be kind of a way of potentiating immunotherapy, because they do an experiment where they combine these NRF2 inhibitors with checkpoint blockade, anti-PD-1 therapy, and this reduces tumor burden compared to not inhibiting the NRF2. And so basically, yeah, another way. I think the idea is that you are, um, by targeting this regulations of the oxidative stress, you can influence the site of the fate of the monocytes and restore the immunogenicity of the monocyte derived macrophages in the tumor microenvironment.
Interesting. Do you think there's any study anymore that doesn't take whatever target you're inhibiting and then do it on top of a PD-1 blockade to see if it's even better?
Yeah, no, because it makes sense. I think the idea, I think it makes sense to combine combinations, because it's unlikely that one intervention will do the trick. And you get, oftentimes, yeah, you get so much more improved responses by combining these treatments. And I think immunotherapy, like checkpoint inhibition, definitely benefits from additional um therapies on top.
Yeah, no, I mean it's obvious that we do it now, but it also seems to be like the trendy thing you always do, right?
Yeah, I mean, it is trendy. I guess it's trendy because I don't want to say it works, but
Well, it
yeah,
doesn't in clinic yet because nothing,
yeah,
but it's like, oh, you got to do it because if you show it, then you have a case for putting it in people.
yeah. That's the first step, isn't it?
Yeah. All right. Well, we're going to be talking in a moment here to Dr. Harry Wong from one of the institutes we just had on the listen to today. In fact, a lot of the
Hmm.
stuff today was from Harvard institutions or Boston in general, which is, I guess, too surprising. But you know what? Brenda? You work with human immune cells, don't you?
I do. I
And you
do.
have to isolate them, don't you?
I have to isolate them, yes.
Well, did you know you can learn how to isolate highly pure immune cells and efficiently process human samples with a free on-demand course on human immune cell isolation? You can learn more and register at stemcell.com/human immune course.
So now for the second part of today's episode, we are very happy to have with us Dr. Harry Wong. He is assistant professor at the Ragon Institute at the Massachusetts Institute of Technology, MIT. And I'm very excited about our conversation because we did cover his latest paper in Cell, talking about some of the kind of nitty gritty details of regulatory T cell functioning. And I thought it was such a cool paper. So I'm so happy that we have you on the show to talk about that a little bit more. So thank you for joining the Immunology Podcast.
Thank you for having me.
All right, Brenda, I know you're chomping at the Treg bit.
Yeah. So, so actually, no, what's your science paper? So I misspoke. It's science paper. That's the one from this year, right?
That's the one
Yeah.
collaboratively with Pete
Yeah.
Savage. Yes.
With Peter Savage and Ron Germain was
Yeah, and
also
Ron
your
Germain.
correspondent.
Yeah.
Yeah. So he was your postdoc.
He was my postdoc advisor. Yeah.
Nice. Okay. So yeah. So let's start with that because I'm a big fan of regulatory T cells, as our listeners probably know by now. And I really like that work. I would like to kind of use that as a starting to talk about your research and because one of the things that are regulatory T cells are like multifaceted cells, right? They have all these many mechanisms, and depending on how you're looking, in which kind of context you're looking at the Tregs, some mechanisms of suppression are going to be more important than others. And I think you did such a great job at showing how important IL-2 deprivation and it's in like really in an in situ situation And there is this quote that I like from your abstract. Let me just pull it out because I thought it was really nice, in which kind of the role of regulatory T cells in this particular situation, in which, for context for our listeners, the importance of antigen specific regulatory T cells in keeping antigen self-reactive conventional cells at bay. And so there was this quote from your abstract, was the model in which you studied, that maybe you can talk a bit about more, complements and advances classical paradigms of self non-self discrimination, illustrating how the adaptive immune system operates on a knife's edge between effective pathogen control and the risk of autoimmunity during infection. And I really like that quote, and I really like to use that as a starter for this discussion. Maybe you want to give our listeners a quick overview of this of your recent research, and how did you look about this balancing out between keeping autoimmunity at bay but allowing for effective pathogen control, and what happens when those mechanisms don't work properly?
Yeah, definitely. So that particular story, I started late into my postdoc, actually, when I was still with Ron Germain, and I connected with Pete Savage, and we started this wonderful collaboration. And the reason we got into that is because Pete actually had this view that the specificity of regulatory T cells should really matter for their ability to control autoimmune T cell responses. And I actually had this view that it should not matter at all, just based on some calculations we had done and known precursor frequencies of T cells. And I think what we ended up finding in the end was that both of us were right. And we went on to show that the specificity of regulatory T cells probably doesn't matter under homeostatic conditions, right? When the system has not been pushed or challenged in any way, any regulatory T cell that's localized to a conventional cell that's been activated by self seems sufficient at constraining the response. But through years of discussions with Pete, they had engineered this system where you had a pathogen that also co-expressed a self-peptide. That sounds very strange, but you can think of it in two ways. One is you can view that as a model of epitope mimicry. But another way to view it is that whenever you have an infection, you're going to have tissue damage and release self-antigen. So the self-antigen levels will rise. And there we found with that model, when you push the system with inflammation and a rise in self-antigens, if you don't have regulatory T cells that are matched to the have matched specificity to that self-antigen, and are matched with the conventional cells, you're in trouble. You will end up with autoimmunity during infection. And that was for a single peptide. And we found that remarkable because if you got rid of that, if you got rid of regulatory T cells specific for that one peptide, you got autoimmunity during infection. It was just one single specificity gone, and you get disease. It was really remarkable.
So how did you go about solving this question? I know you have a pretty sophisticated deletion system.
Yeah.
But you also have some really cool microscopy, which is really what I'm trying to get at.
The sophisticated mouse genetic systems were developed by Pete. And we thought it was a great opportunity to use those models that allowed us to co-visualize a regulatory T cell and a conventional T cell with the same specificity using imaging. And so we used our advanced multiplexed microscopy tools that allow us to resolve individual cells at with reasonable multiplexing capabilities and track their phenotypes to see what happened. And without a challenge under homeostatic conditions, the results were, I guess, unimpressive. There was no selective colocalization between these specificity matched regulatory T cells and conventional T cells. And I remember when then Pete's had engineered a system where they could selectively ablate the regulatory T cell with matched specificity, and nothing happened. And I remember thinking, aha, I was right. Specificity does not matter. They don't localize. There's no functional effect in the animal. They don't get autoimmune disease. And then they came back with this system and said, well, let's try this bacteria that overexpresses the self-peptide. And when we did that and went and imaged it again, now we saw something different. We started to see selective colocalization between regulatory T cells and conventional T cells of matched cell specificity. Not always, though, but we would see it. And to getting at the quote earlier about the knife's edge, what I found was so interesting, at least from my perspective, was there were still some of these self-activated conventional cells that were not surrounded by the matched specificity. They were surrounded by polyclonal regulatory T cells of unmatched specificity. And there, in rare cases, the polyclonal cells seem totally fine at constraining the response. And in other cases, it seemed like you needed the specificity matched ones. And in other cases, you would see breaks in control by imaging. So it's very local. It's not globally happening across the lymph node or any given tissue. It was really like on a case-by-case basis, the system was trying to control the response. And why that variation exists, I mean, there's some degree of stochasticity in the system, but we thought maybe there's just heterogeneous presentation of the amount of self-antigen across the antigen-presenting cells. So by chance, you might land on an APC that has more self-antigen, and that pushes you across the threshold where now polyclonal regulatory cells of unmatched specificity just aren't good enough. You exceed TCR signaling strength and can override the regulatory T cells through some other mechanisms. But yeah, the microscopy was really central for us to really visualize that heterogeneity and understand that it's not an all or none effect, right? That the system still finds a way to win sometimes without the matched specificities, but having the matched specificities is important for some of these isolated events. And if you don't have it, you break control very locally.
So you mentioned that this could be illustrative of a situation of molecular mimicry, for example. We do know that there's a bunch of autoimmune diseases that seem to be triggered by an infectious episode, but not everybody gets autoimmunity after these infections. So there must be certain, case by case differences. Do you think that somehow the repertoire of regulatory T cells or something around the ability of antigen specific Tregs to prevent this has to do with the triggering of the disease?
Yeah, that's a really good question. Let me think about that for a second. I actually think that some transient breaks in control are inevitable with any type of infection or immune challenge that you're going to have, that the system is not so perfectly designed to 100% selectively constrain all highly self-reactive conventional T cells. And I think the difference is in the extent of that response and the extent of escape that you have, which is going to be biased by if you have matched regulatory T cells of matched self-specificity, but also how long it's sustained for, how long the escape is sustained for. And so I think if you're a relatively healthy person and you have an infection and you end up with a transient break in tolerance, you might get a little bit of tissue damage. You'll send a small wave of effector autoimmune conventional T cells into the peripheral tissue. You take some damage, and yeah, that's a bad outcome, but the system has sort of decided that that's acceptable because we need to eliminate the pathogen. And if you're healthy, you'll repair that damage, right? And I think that this is probably the case because, as I've learned from many clinicians, most of the time when you get an infection, you see signs of autoantibodies for like weeks after the infection, even if you're going to recover. But I think the issue comes if you have a slight break in, or if you have a slight bias in your control, or maybe you're a little bit worse, you have a genetic variant where you're not so good at repairing tissue damage, and that this type of insult is sustained. So you have repeated bursts of this happening. So multiple infections and multiple waves of these small cohorts of cells escaping control. Now you get into issues, right? So maybe you're 30% less good at repairing your tissue damage. And eventually the tissue reaches a state where you just cannot repair effectively. And now you're in trouble. Now you have full-on autoimmune disorder.
So it makes sense, right, that you want the Treg keyed up, because too much bad, too little bad. And maybe you're doing that in part by generic versus brand name, as it were, T cell or responses. Do you, from your work, do you have any sense of then where things go wrong and all the people that don't stay healthy?
Yeah. I think it's very possible. I mean, depending on how much you believe a lot of the genetic association studies, a lot of the variants associated with autoimmune disorders map to non-coding regions with selective activity in CD4 T cells, conventionals or regulatory T cells. And a lot of those seem to affect key immunosuppressive mechanisms like the expression of CTLA4 on regulatory T cells, or the high-affinity IL-2 receptor, which presumably would affect your ability to consume IL-2. And so I think if you have combinations of these variants that make your regulatory T cells 20, 30% worse, and then maybe you also have some genetic variants where your tissue repair machinery is 30% worse. I think these things together sum probably non-linearly, because there's so many non-linearities in the system, and that gives you disease. So I don't think I think for a lot of, I mean, certainly there are autoimmune disorders that are caused by a single complete loss of function in one particular molecule. But I think a lot of those, more generally, the disease emerges from a combination of these subtle imbalances that then converge on some nonlinear step that pushes you over the edge.
So going back, so kind of circling back to the way that in which you visualize this thing. So I remember you mentioned first your cell paper from a few years ago, which you have this really nice kind of imaging, which you look at phospho-STAT 5, so STAT5 phosphorylation, which is a signal of IL-2, signaling. And that's how you show, in this case, you're not looking at antigen-specific regulatory T cells, but you do show that the proximity of the Tregs to presumably self-specific conventional cells dampens this IL-2 signaling, and that's what keeps a full-blown self-response away. So do you think? So I think this highlights the importance of really like an in situ study, and really looking at in this case, you were looking into secondary lymphoid organs. How it's really hard to model these things in vitro. So you did a lot of mouse work. I think if you want to look at human samples, it's a bit harder. But how can we look at these very complex interactions to make sense of this particular interaction that really require a spatial organization that is really hard to mimic in vitro?
Yeah, so it is very hard to mimic in vitro, to follow up on your point, because we typically don't know antigen specificities, right? And it's important to know that, because most of the artificial systems you would use in vitro to trigger the T cell receptors, the strength of signal that you receive is way beyond anything that you actually see in natural settings. So that makes it very hard. So I agree, that's why we've invested so much in being able to study these things in situ, where the specificity is what it is. I don't have to know what it is, though. I can sort of be antigen agnostic, and that's sort of the approach that I've certainly taken. But I think, yeah, the ability to track, to actually more generally study the length scales of communication between different immune cells, which they can communicate through physical interactions or by releasing diffusible signals like cytokines, and being able to track that and using something like the phosphorylation of STAT5 to sort of infer statistically how far a cytokine likely diffused in space from a static image. I think that has showed us how much non-genetic cell-to-cell variation exists in the immune system. So, cells, regulatory T cells, for instance, that are positioned very close to IL-2 producing cells are seeing very different concentrations of the IL-2 than regulatory T cells positioned very far back or more distal. And we've shown that, and others have shown that those regulatory cells that are positioned close to the producing cells will adopt very different functional states or differentiation states than the ones positioned further back. So I'm not sure I fully answered your question, but I think that these types of approaches that allow us to actually look at communication between the cells are beginning to explain why you even see so much heterogeneity in functional states and differentiation states in an immune response, right? Every time you do a single cell sequencing experiment, you see, endless cell types and cell states. I think a lot of that, or at least part of that, is explained by where cells are positioned in gradients. And so I think this is one of the approaches we've really pushed on in the lab, for not just for IL-2 and phospho-STAT5, but for different cytokines like interferon gamma, IL-10, IL-6. And we see that different cytokines have very different length scales of diffusion in different tissues, actually. And so this changes how the immune system can propagate and what cells can be involved in the response. But I think those types of approaches are becoming essential for really understanding quantitatively how immune responses or autoimmune responses progress or how they're constrained.
Yes, you talk about cytokines on gradients and being able to measure that, presumably spatially with microscopy and all these cool things. How are you guys able to get around some of the light limits, to do the work you're doing, which I know everyone in light microscopy does it a different way. So could you share your way?
Yeah. Well, so first I'll say, because a lot of the events that we care about, not all, but a lot of the events that we care about simply require single cell resolution. They don't necessarily require us to break the diffraction limit of light. And so that actually makes a lot of the traditional challenges of, very fundamental microscopists much easier to contend with, right? We don't necessarily need, for most of the things that we look at, nanoscale resolution, right? We don't need to be able to resolve individual receptors on individual cells, which is something, I did in my PhD. But what we do need to be able to do is study the immune system as a system. And what I mean by that, it's not one cell type, it's a collection of cell types. So we have to have some minimal degree of multiplexing capability so that we can resolve not just a regulatory T cell, not just a conventional T cell, not just an antigen presenting cell, but at minimum all three of those things, because they all function together as a cohesive unit. And then we need to be able to resolve their functional states, right? So that requires more proteins. So I think the problem of a typical microscopist that's in biophysics or cell biology, which is I need to be able to image 100 nanometers or 20 nanometers, I have to break the diffraction barrier, that there are some questions where we do have to do that. But for a lot of the things we don't. Actually, it's much more about our ability to resolve more and more features in the tissue. And for that, we've invested a lot in methodologies that allow us to multiplex more, right? Use more unconventional antibody fluorophores that are not typically used by microscopists. When I started my postdoc, I'd come from a cell biology and more biophysical background, and at that time there was a limited repertoire of fluorophores that you would use for microscopy. But then when I discovered immunology, I saw that they were using, if you think about a typical flow cytometry experiment, there's so many long Stokes-Shift dyes that you would use, or tandem dyes, or dyes that you would never use in cell biology. And I think for many of us in the field, we started thinking, well, why not? Why can't we use some of those fluorophores for imaging the immune system? And that's what we ended up doing.
Have you had to fiddle with the microscope to get those fluorophores to work?
Certainly, almost certainly. And I will say that the reason we're able to multiplex, at least in my group, so well is because we have a specialized system that has a white light laser that's totally tunable from 440 nanometers to 790 nanometers. So we're not using fixed laser lines like you would on a typical microscope. And we don't use, not to get too technical, but we don't use fixed, dichroic filters. So the system has a special type of beam splitter that where you can change what wavelengths of light are detected on different detectors down to like 10 nanometer band passes.
Wow.
And then without having to swap in and out different filters. And so that has allowed us to achieve, I think we're, a member of my lab has been able to achieve, 15 color imaging in a single shot, right? No iterative immunostaining or anything like that. And then when we couple iterative approaches, which I will say are very labor intensive, at least the way we do it, but are necessary sometimes, then we can get up to 20, 30 colors and beyond type thing.
When you go on those 14 or even 10 colors, are you having to shoot the laser multiple times at the same tissue? Right. So it's obviously you're gonna have to hit it multiple times. Do you guys have issues with bleaching or tissue damage? And have you had to build these? Have you had to take a standard microscope and hot swap it like a hot rod to do this?
We have not had to do too much custom tinkering in house. We've worked with the manufacturers we've worked with them to get maybe some custom things on our system, but we haven't. I don't have a lot of resident optical I don't have optical physicists in the lab that are tinkering. Although I would like to hire someone for that purpose in the future, but yeah, that's really what we've been pushing on. And every time we add a new fluorophore in that we haven't used in or nobody's really used to image, it's a lot of effort to figure out how it's going to fit in and if it's going to work. I mean, the one thing that you may be familiar with is you look at the theoretical spectra, the excitation and emission spectra, and those are made in solution often. And you think, okay, perfect. Theoretically, this should work perfectly and I should be able to fit it in and no issues. But then you go into the tissue and it's not what you think it's going to be. And so every dye is unique.
Yeah, that sounds like a lot of work and a lot of know-how for getting this kind of data, like a spectral microscopy situation almost.
Yeah. Yeah, I mean, it is a lot of work. Obviously, we have a pipeline for this now, and that's been developed over many years. And my lab is, the people in my lab are exceptional, so they've put a lot of work into figuring out how to do this. But that's what we found. That's what it's taken.
So one of the things that I think a lot about regulatory T cells and the importance of the antigen specificity for their function. And when you have access to models like this MJ23 model, in which this what you used from Peter Savage Labs that's recognizing this prostate antigen is really really nice. You can make transgenic mice, but of course when it comes to understanding the mechanisms in human regulatory, it becomes harder. Also because there are differences that we can already see ex vivo or in vitro in how these Tregs from mouse and from humans. So, what can you tell us about where do you think there might be some challenges in translating this understanding of Treg function to the human situation? And if you have like unlimited resources, how would you go about looking at this kind of really spatial behavior of regulatory T cells?
Yeah. Okay. Great question. So I'll give you my honest view. Of course, the mouse is different than the human. Like there are some genetic differences, but my view has always been that this is actually, the mouse can be turned into a strength. I think we have to recognize that the standard mouse that most immunologists use in the lab is the C57 Black 6 mouse. This is a single inbred strain, and it's housed under highly sterile conditions, specific pathogen-free conditions. And the immune system of that mouse basically resembles a neonatal human immune system from 0.00001% of the human population, right? So yes, of course there are differences, but I think at least a significant fraction of the differences come from the fact that we're using the wrong initial conditions for the mouse. So a mouse is not a mouse, is not a mouse, right? If you take a C57 Black 6 mouse and compare it to even a bulb C animal or a B10A mouse or any of the other widely genetically diverse mice, so thinking about the diversity outbred mice or the collaborative cross mice, those are going to be totally different. I mean, even things that we take for granted, when you think, okay, regulatory T cells are 8 to 12% of CD4 T cells. If you look in genetically diverse mice, that's actually not the case. I've seen measurements from like 4% to 25% under homeostatic conditions. So that's my first point. My second point is I think the mice are way too clean, right? A human, when they're born, is exposed to almost instantly to this natural microbiome, right? And that shapes the immune system. So I think when we use these very sterile animals, not that there's not good uses for them, but I think if we think that that's going to be the translating agent to the human, I don't think that that's ever going to be the case, right? And I think when people have used genetically diverse mice or have used wild mice or standard lab mice co-housed with pet shop animals, and you immunize or you infect and you look at the distribution of responses that you get, let's say the antibody titers or the types of antibodies, this is an example, I think you start seeing it looks a lot more like the human population than if you just study the C15 black 6 mice. I make an analogy to saying it's the equivalent of studying my immune response following a vaccine or an infection and thinking that that's going to reflect the entire human population. That's not going to be the case, right? My genetics and my environment are very unique than yours, right? So I think that yes, there's differences and there are some, certainly, I think, what does Mark Davis say? There's 40 or 50 distinct genes in the human and mouse immune system. But I think a lot more of the differences are coming from the wrong initial conditions. And I think that can be, and there are massive efforts to deal with this now, right? People are using more genetically diverse animals. People are using dirtier and dirtier animals, right? And I think that will help with the translating. On the actual human front, though, how do we start directly studying the human market? That's a really good question. I mean, with imaging technologies, it's tough because you have to get tissue from people that have passed away most of the time, or unless it's going to be a skin biopsy or something. So there are certain tissues that are accessible, right? Where most people are saying, oh, I'm comfortable giving you a skin graft or something like that. But most people, healthy people, are not comfortable undergoing surgery and giving you a lymph node or giving you part of an organ, right?
They're kind of needed, right?
Right. So I think there's huge efforts on this too. I think really trying to reconstitute certain organs, organoids for lymph nodes, they're in their infancy right now, but I think that those are going to develop over time. And I think we will figure out ways of controlling the antigen specificity or engineering ways where we can mix in defined combinations of antigen specificity and study their response. So I see I think that's going to evolve a lot in the next 5 to 10 years.
Well, I think we may have to have another conversation in a few years to see where we're right. But we'll have to wrap this up. So speaking of evolution, if you could evolve and pick up a hobby that you don't currently have, mental evolution, bridge or segue, what would it be and why?
Okay, so something I've very much been interested in for the last 10 years or so is picking up a martial art. And not because I'm particularly interested in fighting. I have no interest in fighting, but the discipline and the coordination required to in something like Muay Thai, like actual striking, or in Brazilian Jiu-Jitsu to control people on the ground. I think there's a lot of intellectual thought required and a lot of coordination and a lot of discipline. And I don't know, something about that's very appealing to me.
I can confirm. I used to
you got him started.
Brazilian Jiu-Jitsu for years and striking and kickboxing. I will say, as you do more and more science, I would favor BJJ over Muay Thai from the concussion risk.
Got it.
Getting punched in the face once or twice is a good life lesson and teaches humility. But then you don't need the lesson after that, is what
I'm not interested. I don't want to actually fight. I just want
but like getting hit in class, it's a good life skill lesson to this is what pain feels like a little bit. I'm learning about myself here.
But you gotta protect the thing inside.
Right. Jiu-jitsu is much more sustainable long-term. And there's a bunch of places up where you guys are at in MIT that are excellent.
Okay, I got to check it out.
Yeah.
Awesome.
I mean, that sounds kind of fun, I have to say. I've never done martial art. My brother does taekwondo, but he really likes it. I do think he also liked the discipline and the mental work around it. I think that is very appealing, and that's something that sounds nice to
Yeah, and I think one thing that's, you can speak to it better than I can, but I think some of the sequences or moves, you have to be so focused that you can't think about anything else. So it forces you to clear your mind. You can't think about science while you're doing that. So it becomes meditative on some level.
You got to learn how to have a flow state.
Yeah.
Your brain can be useful because you remember things, but then it can be annoying because you're thinking about your experiment at the same time.
Right.
But my favorite thing to call jiu-jitsu is involuntary yoga.
Yeah. Okay. Got it.
Well, on that note, so thank you so much, Harry Wong from MIT and Ragon Institute for joining the Immunology Podcast. And finally, got to talk about this paper, which I really like. So I'm so glad that we got you on the show. So thanks again for joining.
Thank you for having me. Yeah, it was a blast. Thanks so much.
All right. Thank you.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on X at Immunopodcast, Blue Sky at immunologypodcast.com, or via email at info@immunologypodcast.com with feedback or if you want to suggest a guest. See you next time.